Good evening, Bagaetsho Dumelang, Molweni. Welcome to this inaugural lecture. A very special welcome to our guest of honor, Professor Muthama Muasya, and his family and friends. I want to thank the special people in Professor Muasya's life for sharing him with us at the University of Cape Town. He has worked hard since his early years to develop his field of study and has built a solid global reputation based on his impressive body of work. But an academic's excellence also depends on the support of loved ones. So tonight is also a celebration of the part you have played in his successes as friends and family. It is my pleasure to introduce you to the members of the virtual platform this evening. In case you don't know my name, I am your host, the Vice-Chancellor of the University of Cape Town, Professor Mamokgethi Phakeng. The Dean of the Faculty of Science, Professor Manu Ramutsindela, will introduce tonight's speaker, Professor Muasya, and Associate Professor Tony Verboom, the head of the Biological Sciences Department, will deliver the vote of thanks. 5 years ago, the International Geological Congress, which was meeting here in Cape Town at the time, considered a recommendation that a new geological epoch should be declared. Scientists eventually agreed that the new epoch would be called the Anthropocene, the period in which human activity has been the dominant influence on global climate and the environment. This new epoch dates from the 1950s, when human beings began dispersing radioactive elements across the planet as a result of nuclear bomb tests. Other signals of the Anthropocene effects include plastic pollution, soot from power stations, concrete, and even the bones left by the global proliferation of the domestic chicken, which is cultivated and eaten around the world. Franz Baumann, the former United Nations Assistant Secretary General, calls the Anthropocene the historical period in which the dominant risks to our survival come from humanity itself. The International Red Cross and the World Economic Forum agree that climate change is much more dangerous for humanity than the COVID-19 pandemic. While we have vaccinations against COVID-19 across the world, the only real solution to climate change is to drastically shift the human behavior that is causing this global catastrophe. One important front line in the fight against climate change is biodiversity. Promoting greater varieties of plants and other organisms in the environment builds up ecosystems and protects water resources, the soil and its nutrients, and the air we breathe. Biodiverse systems are better able to break down and absorb harmful pollutants and to recover from unpredictable events such as storms or droughts. So it is of global concern that the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services estimates that 1 million species are under threat of extinction, and that the World Wildlife Fund says that populations of monitored animals have declined by 68% since 1970. An important part of UCT's mission is to engage with the key issues of our natural and social worlds, and to have a positive impact on our society and our environment through our scholarship and research. Professor Muasya's life work is an outstanding example of how excellent scholarship can contribute to our understanding of biodiversity in African landscapes. I now hand over to Professor Manu Ramutsindela to introduce Professor Muasya. Over to you, Manu.
Thank you very much, Vice-Chancellor. I'm privileged to be part of this occasion to celebrate Professor Muasya's achievement in the area of biological sciences. I always wonder how people came to study the things that they have mastered over time. But for Professor Muasya, the journey began from rural Kenya, where he enjoyed a variety of wild snakes as a head boy. At that time, he began to understand the environment in which he lived. He also began to understand the local plants, the animals, and how this could be used for food, medicine, and other cultural practices. This was the beginning of a journey to understand the East African savanna. What became more important for him was to be aware of the seasonality of particular plants and animals. So these intimate relationships with nature shaped his subject at secondary and tertiary levels and paved the career path that he has followed up to today. No wonder that when he went to Moi University, he studied BSc, and he was influenced by the taxonomist there, K. Muthama, who he later on celebrated and commemorated his work with his supervisor. It was this journey that took him to the National Museum of Kenya, where he was interested in the flora of the tropical East Africa. Coincidentally, in those years, the Rio Summit also recognized the need to bring developed and developing countries into collaborations. And this led to the Royal Botanic Gardens in Kew to work with the National Museums of Kenya. This was the beginning for Professor Muasya to have collaborative relationships with the international experts in the field. And he became more interested in studying some of the top 10 largest plant families that have got 5,600 species. Now, understanding the family relationships of these plant species with the biogeography and the evolution of their traits was a fascinating topic for Professor Muasya. That it led to him coming to UCT and continuing that journey by studying the diversifications of the Cape flora. And he was involved in the research that discovered and named more than 100 new plant species, among which are South African legumes. Now, it is important here to note that the work in the Western Cape on the legumes was important towards understanding the origins and biogeographic structures of the biomes of sub-Saharan Africa. And this work has been shared with national and international experts in different platforms. More importantly, Professor Muasya was one of the 3-person secretariat who organized the 2014 conference, which takes place every 4 years. And this is an aspect of taxonomist, where they used African plants to understand some of the classifications that Professor Muasya was involved with. He has not only been studying and publishing, but he has also been a member of other organizations, including the International Association for Plant Taxonomists. So his scholarship, as you could probably Google or search, use search engines to find out, he is one of the prominent scholars in the area that he works on. And we are proud at UCT in the science faculty to have the stature of Professor Muasya amongst us. And to celebrate his work, we are going to listen to his presentations. It is my pleasure and honor to introduce to you our new professor in the science faculty, Professor Muasya, to deliver his lecture. Thank you.
Thank you very much, Manu, for that introduction. Just to confirm that my slides can be seen. Am I coming through, Manu? Thank you.
Yes, you are.
Thank you very much, and good evening, colleagues, friends, and family. It's humbling to be invited to talk to you this evening, especially considering that to be slotted in the VC Inaugural Lecture series at this disrupted time is not easy. But also, I find it a good coincidence that I'm speaking about biodiversity and African heritage during the week and the month that we're celebrating Africa, and where the theme for this year is Africa's arts, culture, and heritage. For the next 40 minutes or so, please allow me to have your audience as I tell you some of the works that I have done. But while I take credit for the talk and for putting together like a chef who puts a broth together, please note that this work is a product of a team of researchers, students, present and past, and collaborators, and I want to acknowledge their contribution to this. In my talk tonight, I'll touch on three main aspects. I'll touch on African savannas and use some of the things, the features that we see in the savanna, spiny bushes, seafoam grasslands, and underground forests as a way to disentangle the complex origin and distribution of the savannas. I'll look at the Cape flora and especially bring out on how we're using the herbarium, Bolus Herbarium, in species studies and works that we've been doing recently in the Overberg, a critically endangered vegetation type. And then I'll step back and I'll retrospect on my journey. I'm a plant taxonomist by training. I practice plant taxonomy, which is the science of discovery, naming, and classification of organisms. I'm an evolutionary biologist. I'm using tools ranging from use of DNA and other molecular techniques to other aspects to see on the evolution. And my focus is mostly on Africa and in the Cape. But to whet your appetite, I also touched on a topic that someone will say I'm being pompous to talk about, on the evolution of hominins, and especially tease on whether there is support for the savanna hypothesis on the origin of hominins. But to set the scene, let me use this image taken from Kenya's Amboseli National Park, looking onto one of the most recognizable landscapes in Africa, Mount Kilimanjaro. On the foreground, we've got a grassy, wooded environment. We've got antelopes, we've got elephants. If you look close at those trees, you might recognize that they are acacia. They are some of the spiny species in Africa. And if you took a transect to the top of the mountain, you'll go through a belt where there are forests. And those forests are like any other African high elevation forest, like we find in the Cape. Then above the forest zone, you get into a heathy vegetation, shrubby, like you've been on top of Table Mountain with giant ericas and such. And then you've got permanent glaciers. But you might assume that these have been like this for long, except that if you took each of those components I've spoken about, you'll find that antelopes have been shown to have arrived about 30 million years. Mount Kilimanjaro is a baby, only of a volcanic origin in the last 5 million years, whereas elephants have been in Africa for long. So prepare yourself to hear a talk which takes you through space and time and allow for the dimension of years to be millions of years as opposed to decades, please. A lot more in harmony with nature and whose cultures we recognize and celebrate tonight as part of celebrating Africa's heritage. But if we stepped into the past of where the Maasai community are to about 3 million years ago, we find steps of some of our hominin ancestors, Australopithecus afarensis footsteps at Laetoli in Tanzania, and the works by the Likis were important and fundamental in revealing this. And as a young scientist at the National Museums of Kenya, being at the environment where the Likis worked really shaped my stimulation for science. But still, an artistic reconstruction of Laetoli shows Australopithecus walking on 2 feet, giraffes, but very little on plants. So we could ask the question, what did they eat? what was the environment like, and I'll try to answer some of those. Just to give you the big picture, Africa is one of the most diverse continents with nearly 60,000 species of plants. Only South America has got more plants. If you look at those plants, you can put them into major biomes with equatorial forest to the west of Africa, with the savannas surrounding the equatorial forest and more to the east, with this Mount Kilimanjaro and other high elevation mountain and alpine landscapes. And then we've got the Cape at the southernmost tip. And of course, Africa has got two deserts, Sahara being the largest. And it's thought that forest dominated the continent to about 60 million years ago, and that at around 20 million years ago, there's gradual opening. And if you look at the Cape flora, which is one of the hottest biodiversity hotspots, with 9,000 species, many of those species have evolved in the last 15 million years. And the Sahara, for example, is thought to have arisen due to changes on the plates with the Tethys Sea, which separated Africa and Eurasia, closing, and that blocking moist air from flowing through. But how do we make those pompous claims of the past? We could use fossil evidence, and unfortunately, fossils don't form equally, and it depends on where the organism died and whether there was conditions for fossilization. So Africa is poor in fossils. We could use indirect methods. So for example, look at the teeth or the bones of animals and look for signal on the vegetation that they ate, and that has been used. An alternative is use phylogenies and to reconstruct using modern methods the history of organisms. To unpack further on phylogenies, on the left is a famous figure from Charles Darwin's Origin of Species. And here he was trying to think through on how revolutionarily from an idea of a common ancestor, you can have over time modification and the inheritance of traits leading to tips that we see now, having traits which are separated from the other species that are living now, but sharing common ancestry. So if you look at the big three, they have got a thick skin, and that thick skin has been thought to unite them, and they were thought to be closer to one another. But use of more data is showing that the elephant is more closer to the sea cow and the Orochirax. The rhino is closer to the horse, whereas the hippo is closer to the dolphin. And I can tell you that over the last 30 years, especially the use of molecular data has allowed, DNA molecular data has allowed us to refine the tree of life, and that has been part of the work that I've been doing and I'll be presenting. It's a beautiful time to be a biologist. From the COVID virus being seen, it's been sequenced, and within a couple of days you can get a full genome sequenced. The technology has advanced. We've got specimen data scattered in major museums and herbaria around the world to observations on the day can all be put together, and there are global databases that allow us that. There's computational power to allow crunching of huge amounts of data. And importantly, we've got scientists from different countries. Language is no longer a barrier, and also discipline is no longer a barrier. So we are in an era where it's collaborative comparative research. But let's get to the first part of my talk on the savannas. And as I use this scene from Nairobi National Park, looking to the city, where you have a C4 grassland, the whistling thorns, acacia, and rhino. But if you stepped a few kilometers from here, where the soil changes, you are in a montane forest. And Nairobi is at the cutoff between forest and the savanna grasslands occurring. And the work by Mahesh Sankaran and co-workers demonstrated and mapped where savannas are in Africa, showing that they are mostly in the tropics, and that depending on rainfall, if you are above 800 millimeters of mean annual rainfall, you get mesic savannas, especially if there's 6 months of dry season. If there's less drought, then you switch into forest. Once you drop below 650 millimeters, then you have arid savannas, and in gray are areas that are supporting savannas. If you look at savannas, and I use the example of Serengeti and the Kruger in the images, they've got herbivores, they've got grasses, and those grasses are C4. The trees that are there, the bushes are spiny, and fire, especially the mesic savannas, is an important factor, or antelopes in the other savannas. And I'll use spines, C4, and fires to tease on the original savannas. Starting with spiny plants, these are classical seen from many of the savannas. Antelopes trying to nibble leaves between the acacia or other spiny bushes, and the plants having high densities of thorns. And with increasing carbon, those thorns are even getting mega. They're really big. And if you look at what becomes spiny in a plant, it could be a sharp shoot, it could be a modification of the leaf, or it could be the outer epidermis of the leaf, of the stem, that becomes spiny. If you look at where the spiny plants occur, and figure A is showing the vegetation types, we find that B, which shows spinescence, spines are mostly in the savannas. There's an odd spiny plant in the equatorial forest, and that spines are in areas that have got antelopes, the mixed size social feeders, and also the large browsers, but not in the wet forest where we've got the non-social browsers. We've put together a phylogeny, a tree of life sampling over 2,000 woody species from Africa, and sequenced for 2 of the main markers that are used for that level of study, and did a tree of life which is calibrated with fossils. And on that tree, which is shown with the names on the left, we mapped where spines are occurring, and we showed that it's after about 17 million years or so that we got the explosion of spiny as a trait in the savanna in about 25 or so different families. And our interpretation on this is summarized in this figure, where if we look at, I draw your attention to the scale, present being 0, past being 140 million years, there's been a slow accumulation of woody species which have no spines. But the ones in spines, shown in red, seem to arise from 40 million years, and at about 20 million years, they have an exponential increase. That also coincides with when the bovids are increasing. And where we've got high diversity and dominance of spines is also where we've got bovids. I know someone's hand is shooting up in the audience and asking, wait a minute, where did the bovids come from then? And it's thought that the African and the Eurasian plates came together at about 40 million years, allowing interchange, and the ancestors of the bovids coming into Africa and starting an arms race leading to the plants being spiny. Before that interchange, Africa had elephants and members of the elephant group, but not the antelopes. Moving to C4 grasses, and an example of a C4 grass is maize. Most plants use the C3 photosynthetic system. They absorb carbon dioxide. They have to open their stomata to take in the air and incorporate to make a 3-carbon sugar starter. But some plants, 70% of them being grasses, have evolved a mechanism where they actively pump carbon dioxide and combine it into a C4 pathway and are able to close their stomata when they are photosynthesizing within specialized cells, which allows the C4 plants to occur in drier, hotter environments, but not under shade. They need a lot of sun to be able to flourish. Where you can grow maize naturally, basically is where C4 plants occur, and this map in red shows those areas. So lots of the savanna areas in Africa can support C4 plants. But then a study about 15 years ago hypothesized that the C4 grasses, which are occurring where the antelopes are, evolved from an ancestor who was in a wet open environment through the forest. And other groups like the sages went straight to the grasses. And we wanted to test these using more rigorously sampled DNA phylogeny. And when we reconstruct these, we find that, yes, the ancestor was in the open, but was not in wet. And that there are some groups which are from an open habitat went into the forest, shown in green, especially the relatives of grasses. Or there are some groups which went into wetlands. But where we have many species occurring, groups that are rich in diversity is those which have got in the open, not in wet. And importantly, it's groups which have evolved C4 photosynthesis among the grasses. So for example, the panicums, which includes the maize and such. But also C4 photosynthesis system has also evolved multiple times among the grass and grass relatives. including members of Cyperus, papyrus being an example of that. And if we look at the age, we find that they arise from about 30 million years. And this has really puzzled people, especially when the ecologists look at the evidence from the teeth of animals, and there's accumulation of C4 signal in the teeth from about 10 million years ago. There's also charcoal, and there's also those signal of those isotopes in the soil. And the question is, what happened? How comes this group arose 30 million years, but it's only in the last 10 million years that we've had an ecosystem which is C4? And we think fire is the main thing. And I will explain how fire is. Let's look at another trait which is linked with fire, which is the underground forest. I know someone will ask me, how am I contradicting myself? How do you have an underground forest? And the concept of underground forest was coined by Frank White, an Oxford-based ecologist who was working in Africa, and saw that especially in the Zambesian region and into the high fjord of South Africa, members of tree species have got a tree individual, and they have got what looks like a herb, but the herb is mostly coming from a really well-established underground structure, which is called sulfurtex or geocyclic. And this is a phenomenon that we find in Africa. We also find in Brazil, in the Cerrado, which is also an equivalent of our savanna. And if we use the same DNA phylogeny that we did for the woody species, we find that there is multiple origin of these underground forests. And these underground forests are in groups like members of the coffee family or members of the protea and others. If we map using specimens and other data on where they are occurring, we find that they are the oldest being the red and the youngest being blue. The oldest are around the coasts or nearer the equator, and that the youngest as we come further south. So if you're in the high fjord, around Johannesburg, those grasslands will have these underground forests, but most of them are mostly young. Young, I'm talking about 3 million years, as opposed to old, going back to 10 million years. So as a take home from this savanna story, we think that different components of the African savanna have evolved at different times. That spiny plants have evolved earlier, around 20 million years ago, responding to the arrival of the bovids. But C4 grasses and grasslands, as we see them today, are more recent, and those coincide with the underground forests. And also those coincide with when charcoal becomes important or noticed. And we think that it's fire which has driven the expansion of C4 grasses into forming the ecosystem we call grassland. And that fire is not in response to dropping carbon dioxide or changes in temperature, but we think it's more because of the shifts in the ocean currents and moisture. I talked earlier about the closure of the Tethys Sea separating Africa and Eurasia. Within the last 10 million years, South America and North America have come together to join. Also, there's formation of the Wallace Line linking Southeast Asia to Australia and blocking the flow of what used to be moisture from the Pacific through the Indian Ocean over through the Mediterranean to the Atlantic. And also this time coincides with the Benguela current, which brings the cold, wet weather we have now in Cape Town. And there's that shift in rain patterns and aridification that has increased the incidence of fires and allowed grasslands to flourish. But let me walk into a territory that I'm not on solid ground to ask the question on whether we can test the savanna hypothesis. It is thought that the hominins are successful because they were able to walk on 2 feet. And walking on 2 feet is advantageous if you are walking on open ground where you can see further, where you can see your enemies, but also the shift into a savanna allowed plants which had more nutrients for a bigger brain to grow. But these are reconstructions of the environments where the fossils have been found scattered mostly along the Rift Valley down to South Africa. Some of those habitats, like Lake Turkana area in Kenya, have got fossils of trees. So there must have been forests around 14 million years, but now are fairly open. If you look at the hominin family, and it's a big family, and I don't expect you to follow the names and dates, you find that there's almost a trend that the oldest ones, from about 7 to 6 million years are around the equator, and that the youngest are further south, coming down to South Africa. And we used the phylogeny we did for the woody species. And that's the question. If the ancestor was in the savanna, then you should have a savanna-savanna split of species of plants. And if we mapped that on a latitudinal scale, is there support for the oldest savanna splits being at the equator to support, a pattern like the fossils are found? And voila, there's correlation. The oldest fossils from about 10 million years ago are the oldest savanna splits are from about 10 million years ago from the equator. The youngest as you go further north or further south. And this work, we think, allowed us to take another angle into the cradle of mankind paradox. And I tell you, boy, this paper was tossed around from journal to journal. It took about 3 years to get published last year. We were very happy when it got published. And in doing this, we've had lots of fun. We have had a team of researchers collaborating, ecologists like William Board from UCT collaborating with phylogeneticists, different institutions, University of Johannesburg team led by Michelle Vanderbank, bringing Jonathan from Canada, naturalists like the Barrows and Mike Bingham. And this allowed me to almost be in the space that I wish to be in, to ask big questions, working with teams that stretch my knowledge, but allow me to gain more skills. Allow me to move to the second part of the talk, focusing on the Cape. I don't know what's your view of the Cape. Is it the scenic landscapes with the Cape Fold Mountains having breathtaking drops, or is it the provision of spring flowers like our second year students see the West Coast when we go out to teach in September? Or is it the after fire environment with the recent fire at UCT? I'm waiting for the spring to go up there to see what's growing up. But if we put it into context of the region, we find that we can recognize 4 main biomes: the Fynbos biome and the succulent Karoo in the winter rainfall area, the savanna and the grasslands, which are in high elevation in the summer rainfall area. And there seems to be a gradient determined by temperature and rainfall, and in addition soils, in determining which vegetation type is where. But putting together this knowledge on which species are where in a region with over 20,000 species is not a small undertaking. It's taken hundreds of years, and it's taken involvement of an art, the herbarium. One of the unique things of being a plant taxonomist is being able to go into a building and find old pressed plants with the notes from where they were collected. Now the Cape Flats is all buildings, but we can tell you what plants grew in Rondebosch Common or where UCT is viewed from those specimens. And this art of preserving dried material goes back to about 500 years when it was used for documenting medicinal plants. But for the Cape, there's nearly a 500-year history of collecting, with the main collectors who came through, including Thunberg, who was a student of Linnaeus, going through a period when specimens were collected and exchanged for sale in Europe, to the first collection, which is in a South African herbarium. And then more importantly, the University of Cape Town and the Bolus Herbarium has had really determined and focused collectors, especially Else Steyusen. You might think she's short, but she's a giant among botanists in terms of collecting over 40,000 specimens, many of the first set of which is at the Bolus Herbarium, but distributed elsewhere. Our work, mostly from high elevation, is so important that she has over nearly 50 species named after her. I've named a species which grows on rock crevices of the sages after her. Then we've got modern collectors like Nick Helm, who is now looking at fragmented landscapes. Now, focusing further on the Bolus Herbarium was bequeathed to the institution, the University of Cape Town, together with a very rich library by Harry Bolus. It was started about 150 years ago. It's been at UCT for over 120 years. It's got a rich history, being tossed between Kirstenbosch and the university at one time. Pearson, who was the director of Kirstenbosch, was also the head of department of botany. And this collection is is really important for research and teaching. You know, I'm standing in front of a typical cabinet. What you're looking at my front is the typical specimen. We've got about 500,000 of these. On each specimen, glued on paper, archival paper with a label showing all the details on collection. And then the folders that are showing in red are folders containing specimens whose plant names are linked to, type specimens. We've already documented over 15,000 specimens and as part of a global initiative to image specimens. But this is what makes the Ebola special. You can get DNA from them, you can get chemistry on them. Our ecology colleagues are looking at shifts in plant traits due to increase of carbon dioxide, and it's a minefield for future uses. This collection is associated with a curator who has 2 staff I work with, 3 academics, my colleagues Tony and Terry. Terry working on Moses, a group that I don't touch, but where there's plenty of diversity. We've got researchers, honorary research associates, Peter, Charlie, and Nicola, also working on other groups. And then we've trained students over time, Zainab, Daniel, Tammy, Abu, who have come through in the last 10 years and contributed to our knowledge. It is this valuable collection that we treasure, and one which last month when we had the fire at campus, and when the fire caught onto the botany building, the 2 ends, we panicked, we prayed, we got calls from local and international collaborators, and we are really, really thankful. If it was a physical audience, I would have said, let's take a minute just to applaud those people who saved this collection and who ensured that it did not burn. You know, we had water damage, which was also quickly attended to. We are committed to get this resource stewarded into the future. We already have an initiative to image these specimens and to database, and we will be making follow-up on how we could speed this process, because there might be another fire soon, but also this important collection needs to be available to the rest of the world. But linked with the bolus, in the last 10 years, there has been really interesting taxonomic work. We've described over 100 species, 7 genera, and here's just a quick breakdown of the plant groups which they have come from. The peas, fabaceae, legumes, especially asparagus, well, Lauretifer rooibos, or Sorelia and Ortholobium make about 50 of those species. Cyperaceae, the genus Chinus, and Phycinia also make about 30 species, and then other groups. But not just the taxonomic work of describing species, we've also been involved in monographic studies. And a classical one is work by Peter on the Staphylians, which won one of the best book awards a few years ago, and where he's worked with a team looking at this group globally. The bolus is making a huge footprint on the taxonomic studies of the Cape and of the world. Many of our plants are only here, but we've got some which are here and elsewhere. But as we do this, we found that some of those 10 species that we've described have come from the Overberg, an area that you can drive through less than 50 kilometers at the widest, in between the farming land. And as bigger machineries are made, we're finding that the patches of Renosterfeld, a unique vegetation type, that are remaining and getting smaller and smaller. And we've got these quartz islands, rocky, which the farmers have left because it's either too rocky or too steep. And these are the habitats that are turning up unique species. There are up to 25 currently known narrow-ranged species that are only in this area, not more than 50 kilometers wide. Within the general Eastern Overberg, there are over 70 red-listed species, species that have conservation concern. And this then says that we cannot go and use the rhino approach to save one species. We've got to have an ecosystem-based approach to conservation if we are to secure not only the species that we're looking at, you know, Asparagus quadricolor, only described 8 years ago, but also the bacteria that is linked with for it to fix nitrogen and to live. So we need to secure the environments in totality. This, thankfully, has been done by a champion of the Overberg Renosterfeld, Odette Curtis, who did a PhD in our department, and who is an honorary research affiliate of our department. And she's been working here since her PhD studies, and seeing the special nature of this area and being part of that initial study, worked with conservation stakeholders to purchase one of the largest patches of Renosterfeld in Everscliffe. For those who go to the Hoop Nature Reserve, it's in that area of the southernmost tip of Africa. And she set up a foundation, a trust, Overberg Renosterfeld Trust, Conservation Trust, which is fairly small. You know, her and 3 other people, 5 trustees, of which I sit among. And with that, being able to bring together scientists to respond to questions on what is where and why is it there, and questions on what's the impact of grazing on this, or what's the seed recovery, what's pollination like? Are we conserving a system where there are no pollinators? And auditors being able to engage academics from UCT, myself, several others, members of my department, other departments, other universities, and other specialists into answering these questions. These are an area that we're using to train the next generation of conservation biologists. But at the same time, she has been able to bring in the farmers and to tell the farmers, give them feedback on how special these fragments that are sitting in their land are. And farmers are coming in to say, I want to eke a living from the land, but also want to conserve it to perpetuity. For example, Seine wines, and they make very nice red wine, have put part of their land to conservation. Also, last year, we celebrated the publishing of a new guide, field guide, on the Renosterfeld. So making impact on the knowledge and knowledge production. But we seem to have more and more farmers coming in. These are 4 others who have come in the last year. And we think that this model of working with the farmers to secure fragmented pieces of land on their own private land perhaps is the future of conservation, as opposed to huge nature reserves. Anyone who has visited a farmer to access their land will know that it's not an easy undertaking. It takes lots of patience, lots of cups of tea, coming back, and Odette has that patience. Well done, Odette. But one of the species that we described from the coarse patches in the Renosterfeld, Overberg Renosterfeld, Schinus quaesiticus, is of a group that we've been revising its taxonomy. It's a group that is in South Africa. It's a group that's in Australia, with about 40 species in South Africa, with about 100 species in Australia, 1 or 2 species get into Europe. And it's one that we've been understanding its evolution, hypothesized that it dispersed as a single event to South Africa and then has made more species. But bringing its taxonomy has required really careful, painstaking work, because the genus Schinus and Tetraria were both describing or based on types from the same lineage, and yet some plants in Australia called Schinus or called Tetraria are actually not members of this group. So it's taken sorting the taxonomy to put what was put out of Schinus in South Africa, what should not be in Schinus out, and especially Russell Barrett, Karen Wilson, and Jeremy Bro in Australia have been part of that team. And then describing new species, sorting species names. We've had to, things that were double named have had to be synonymized. This, to me, is one of my biggest contribution to science of taxonomy in my career. But as part of this, and last year during the lockdown, when we were in doom and gloom, we described the species from Tokai. You know where those pines fell, pines must fall. That area being restored, a species that there was a fragment of specimen from about 60 years ago. We were able to find a population and to name it, and the name tells it, Schinus inconspicuous, not very conspicuous among the grasses. But 2021 to me and to my colleagues has been dubbed the year of the sages. It's a year that an international community of collaborators have come together to put all our data and to use all the available tools to do a new classification and taxonomy of the Cyperaceae. And this work led by Isabel Laridon at the Royal Botanic Gardens Kew, and bringing together about 50 co-workers, has had a seminal output, which is coming out later this month. But also thematic papers. For example, the Schinus work went into this. But as part of that, I contributed also to a savanna group, Fibristylis albigardia, where we had 2 species known before I was born, which have been tossed between albigardia and Fibristylis because their morphology could not be told unequivocally. And the DNA data tells us that they are not albigardia, they are not Fibristylis. Actually, they are far removed from them. And if you are to put them into Fimbri Salis or Abigadia, you've got to merge the whole group. And the decision by the international group was to keep them separate. And I had to form a new genus. And I had to find a name for that genus. And I decided to celebrate the Zulu community, to use an African name celebrating a community in Africa as part of our taxonomy. Partly because these two species are in KwaZulu-Natal mostly, but one of them goes up to Tanzania. But making a conscious effort to be representative in my science, in how I use and I name things. Take home, if we look at the Southern African flora, there's complex interchanges. We can recognize 4 main biomes. Of these biomes, if we look at the Fembos, there's more species dispersing into the succulent Karoo than back. But if we look at the continents, we also find that there's been lineages which have dispersed to or from Australia, and the interchange between species between the continents happened between from 60 million years to the more recent ones before humans came on the scene. For some reason, there isn't as much interchange between Africa and South America. Please indulge me in the next 4 minutes or so to take a step back and reflect on my journey. And this is where I grew up. My weekends and school holidays were spent herding the family herd of goats and cows in the Mesic savanna, spines and such. Tell a current adolescent male to spend a day following goats and they will go on a riot. But I had to do this the family income. This is where my fees, school fees came from. These are the animals which were part of our meals and our milk. Some of these animals also I've used to exchange for getting the beautiful wife that I have. But I feel privileged that I've had opportunities, because many of my contemporaries who were herding together are still in the village. It's a good life, but I have been able to do a lot more things out, especially because of a number of factors, which I celebrate. One is of my family. My parents are the generation that went to war. My father went to the Second World War. He doesn't know why he went as part of British troops. Coming back, finding a wife that had been found for him in case he doesn't come back, and having 9 of us, 8 of us living. And my parents were unique of parents that sent girls to school at a time when it was considered a waste of your cows to send a girl to school. Talk of patriarchy here. But being the last of these 8 living children, I had the privilege of my siblings helping my parents to look after me. And also that I did not have to stop school to earn a living. And I really, really thank each one of them for that, including passing down a shirt when I needed one. Also, over the last 30 years, I've had a friend who's been a field assistant and my pillar of support, and my wife. And we've together recruited 2 more assistants. So the team is even bigger. Thank you all for your contribution. But taking from the time when I finished my master's degree and went to National Museums of Kenya, as Manu introduced, it was a time when the Global Environment Facility was building taxonomic capacity, especially in biodiversity-rich countries. And my colleague Cyril Masinda and I were sponsored to go to Kew Gardens to work with experts on our plant groups. That process and that mentorship involved Eric Knox, who was deployed to Nairobi and who has remained a friend and a collaborator up to now. And also we had contact with Charlie Staton when he was the Deputy Director Research, Kew. And actually between Charlie and Eric, while camping at 4,000 meters on a trip in '94, my PhD concept was conceptualized. And that's how I ended up in Cape Town as a student to collect, because I was working on plants that are on Mount Kenya, Kilimanjaro, but also in the Cape. But going to Kew as a PhD student, I found 2 very generous and really impactful academics, actually 3, but here Dave and Mark, allowing me to develop molecular skills at the general, at the same time the skills at the herbarium, and the contact of being at Kew, that was really priceless. And both of them still remain as collaborators and friends. And then after my PhD, I got an opportunity for doing a postdoc in Leuven in Belgium with Eric Smets. and to work with the world expert, Paul Kutubia on cyperaceae. Again, they've remained collaborators. And this, I would say, among the people who have shaped my journey more recently. But there's lots of other people who have assisted me. You know, I rocked up in Cape Town in '97 without a driving license, and Tony and others had to drive me to the points that I needed to go to. Thank you, everyone, for your support. This has allowed me to come to UCT later, to become among the community of scholars, to be able to mentor postgraduate students. This is a group that Samson and I had and still have an image from before COVID, but also looking at those students, I'm proud that I can celebrate Africa Day and say I've contributed to capacity in Africa. That investment done in me has multiplied. And that experience from research has also impacts on my teaching of my first year class to second year to third year, and also my taking into the global community of practice in my field. If I can use the words of the African theologian and philosopher John Beatty, I am because we are, and since we are, therefore I am. I've drunk and enjoyed the wisdom and knowledge from others, and I'll freely share it. And also I ask my students to do the same so that we can have the goodwill seed planted and multiplied among us. Among the communities that I also spent time with is one of UCD's largest residencies, where we've got 900 students. And these are senior undergraduates and postgraduates. This is a space where we want to build academic excellence. This is where we want to develop leaders. This is where we want to confront issues facing society. And boy, it's a fun and challenging space, but also one which is very rewarding, especially when you think that this is the future of Africa, and be able to confront issues of patriarchy, dealing with challenges, you know, the recent fire and all that, has been extremely rewarding. If I had another life, I'll still give it to this enterprise. So thank you so much for 40 minutes of your time, for allowing me to take you through my passion, taxonomic research, evolutionary research, to showcase some of the species discoveries that we've done and taxonomic work, and to talk about the savannas, and to allow you perhaps to be able to confront some of the falsehoods. For example, the image of Simba here, which is supposed to be in the savanna, but if you look at the top left, they are in an Afro-Alpine zone. They must have been freezing. Thank you very much, Asanteni.
And now invite Professor Tony Perbom, the head of the Biological Sciences Department, to deliver the vote of thanks. Over to you, Tony.
Thank you, VC. So in 1995, in between completing my master's and starting a PhD, I spent the better part of a year at the Royal Botanic Gardens, Kew, where I was working as a curatorial assistant in the herbarium there. And during the course of that year, I was introduced to this young Kenyan botanist who was conducting his PhD research under the tutelage of Mark Chase, who at the time managed the Molecular Systematics Laboratory at Kew. And being both from Africa, there was a bond between us, and we struck up a rapport. And little did I realize at that time that we would one day be colleagues in the same department. And indeed, that this young Kenyan would one day be my boss. Well, the young Kenyan botanist in the story is, of course, the same Professor Muthama Muasya, whose interesting career and life journey we've just been hearing about. Having completed his PhD in 1998, which was a landmark work in really providing the first real picture of evolutionary relationships in the sedge family Cyperaceae, Muthama, as we've heard, returned to his native Kenya, where he resumed his position at the East African Herbarium. But determined to improve his skills and CV, he's a very determined man, he pursued a series of international collaborations, including with Professor Eric Knox, who was a major influence. And then later, having taken up a Smuts Memorial Fellowship in the Department of Botany at UCT, Muthama applied for a permanent position in 2005 and was successful, taking up an appointment as senior lecturer the following year. So, having started out as a sedge taxonomist with an interest in wetland ecosystems, Muthama has considerably broadened his research expertise during his stay at UCT, acquiring expertise in the ecology, floristics, biogeography and conservation of savannas, of the karoo, and also of fynbos vegetation. In addition, via an extremely fruitful collaboration with Professor Charlie Sturton, Muthama has expanded his systematics interests and expertise beyond the Cyperaceae into the legume family Fabaceae. And he's now recognized as an authority on the systematics, taxonomy, and ecology of both of these families. And it's worth noting that both of these families are ecologically and economically very important. Most recently, as we've seen in his talk, Muthama has developed a deep passion for the quartz field flora of the Western Cape, and his research on these systems also has a strong conservation aspect. And as is typical of Muthama, his work on these systems has been highly community orientated and has seen him interacting with conservationists, with landowners, also with citizen scientists. And he has infected all of these people with his love of plants. Amazing. Today, Muthama boasts an outstanding research record. If you look at his Google Scholar profile, it reflects no less than 243 publications, 4,300 odd citations, and an H index of 35. But beyond his research contribution, of course, Muthama's service to the university has been outstanding, both as a teacher and as an administrator. At the undergraduate level, Muthama has faithfully delivered a full teaching load each year, consistently enthusing students with his remarkable passion for plants. At the postgraduate level, he's likewise been exceptionally active, graduating 15 MScs and 8 PhDs in his time at UCT. Significantly, his lab has always been a model of human diversity, embracing a mix of cultures, genders, and building capacity within sub-Saharan Africa. As departmental head, Muthama steered the department through some extremely choppy waters. His term straddled the 2016-2017 student protests, the introduction of austerity measures, and the job cuts that entailed, the most difficult years of the Western Cape drought, and of course, the start of the COVID-19 pandemic, all quite significant challenges. Notwithstanding these challenges, Muthama also achieved several important goals that added considerable value to the department and the way it functions. And these include the recruitment of several excellent staff members, a fundamental restructuring of the undergraduate curriculum, and also, and not least, a revitalized program of digitization, which is geared towards moving the Bolus Herbarium into the 21st century. And all of this was achieved while simultaneously, as Muthama alluded to, managing an extended family of undergraduate students in his capacity as warden of Oxford Square residence. Not sure I could have handled all of that. Perhaps most importantly, from a departmental perspective, Muthama has been a level-headed, compassionate and thinking colleague, the kind of colleague really that one is very happy to have battling in the trenches alongside one. And this has been crucial, particularly in recent times, as the department has been forced to engage with a plethora of challenges that quite honestly were beyond its control. And Muthama, I wish to thank you not only for your excellent talk this evening, but also for the enormous contributions that you've made to students and colleagues in bio and at UCT in general. You really have made a very real difference. VC, over to you.
Thank you very much, Tony. And thank you to Muthama as well. On behalf of the UCT Council, Senate, and the entire UCT community, I congratulate you, Professor Muasya, on your professorial appointment, and thank you for your tremendous contribution to our institution, our country, and our region. I wish you many more years of professional success and personal fulfillment. Congratulations, Muthama. And thank you very much to the hardworking team in our communications and marketing department for organizing and managing this virtual inaugural lecture, and also to all of you attending this lecture. Have a good evening.